Hello and welcome to this Simpleware Software customer webinar. Thank you for taking your time out to join us today to learn more about one of our case studies we have. It's called Leaking Gutters, Using Engineering Tools to Analyze Endovascular Device Interactions. The presenter today will be Jacob Kwasinski from the University of Oxford. I'll introduce him a little more on the next slide or two, but just to start with, my name is Ross Cotton. I'm the Application Engineering Manager for the Simpleware product group at Synopsys. And my role today will be simply to help organize the webinar in terms of the presenting and the Q&A at the end. So what we'll do, just to go through the agenda and instructions for today. The webinar will be a presentation by our guest speaker, and we'll finish up with a Q&A at the end. So the Q&A at the end. So if we don't get, we may get quite a few questions. Just ignore the text on the screen there. That's a little incorrect. So we will be doing questions after the webinar. Please do type those into the questions toolbox, and we'll be able to get back to you in the Q&A. If you do have any specific technical support questions, I would direct you to the Simpleware-support email address. That's the fastest way to get any technical support with the software, and just use the questions today for questions on the topic of the talk. Just to make you aware, the webinar is being recorded and will be made available for download. If you do experience any problems during the talk today, please report those into the Q&A toolbox, and we'll try and help you out the best we can during the live presentation. Okay, so just to quickly introduce our speaker today, Jacob Kwasinski from the University of Oxford. Jacob's recently completed his PhD in engineering science at the University of Oxford, working with the Special Structures Group, the Institute of Biomedical Engineering, and the Interventional Radiologist at the John Radcliffe Hospital. His research is focused on vascular disease, experimental and computational biomechanics, and medical device development. He has a bachelor's degree in biological engineering from the University of Guelph in Canada, and has a master's from Oxford in 2011. Also has a bit of industrial background as well, where he worked as a principal engineer and R&D manager as well. So with that, I'll hand over to Jacob. I'll make you the presenter, and I hope you enjoy the talk today. Okay, Jacob, it should be under your control now.
All right, perfect. Great. Thank you, Ross, for the kind introduction, and thank you everyone for joining this morning or this afternoon, wherever you're calling in from. My name's Jacob, and as Ross said, I'm going to be sharing with you today some research coming out of the Special Structures Group in the Interventional Radiology Unit at the John Radcliffe Hospital here in Oxford. And really sharing with you some engineering tools to analyze endovascular device interactions during complex endografting procedures. Now, today the talk is going to be purposefully broad. I think we have quite a broad audience, so I don't want to focus too much on specific details. What I'm hoping to share with you is both some experimental and computational tools that we've been working on implementing here, and I think those in the audience are perhaps most interested in how we used Simpleware Scan IP to really quantify some of the behaviors we're most interested in. So today I'm going to share with you what are these advanced endovascular techniques. Like I said, I'm going to elaborate on some of the experimental and computational tools, and then I'm going to point to two very specific examples of how I use the Scan IP in my work to really quantify some of these interesting interactions. Okay, so a bit of an overview. I'm going to motivate what we're talking about today and frame it within kind of cardiovascular disease, and I'm going to focus in particularly on the treatment of aortic aneurysms, and that is thoracic endovascular aortic repair, or TEVAR, and a subset of TEVAR known as chimney TEVAR. I'll then kind of outline, like I said, the computational framework we've developed, as well as some in vitro and benchtop studies. And I'll then finish things off with a general summary and an outlook. Okay, so those who work across the healthcare industry will be familiar with using things like mortality rates to frame the research problems and questions when motivating their work. So here we see a clear delineation between the causes of deaths worldwide. Now, topping the list, and by nearly a twofold increase over the next segment, is cardiovascular disease. So when we're working within the cardiovascular space and addressing some of the key research questions within, it really represents a significant opportunity to help patients and their families, as well as healthcare systems, in addressing some of these really pressing needs. Now, one particular manifestation of cardiovascular disease is thoracic aortic aneurysms. Now, an aneurysm is a diseased dilation of the aortic wall, whereby if the stress in the aortic wall exceeds the wall's strength, fatal rupture can occur. Now, diseases of the thoracic aorta, whereby the thoracic aorta is the main blood vessel in the body, can occur along its very distinct segments, that being the descending portion, moving into the aortic arch, or those of the ascending aorta most close to the heart. Now, to treat diseases, surgeons have turned to thoracic endovascular aortic repair, better known as TVAR, to manage these patients in a minimally invasive way. Now, during these procedures, a stent graft device, which we see here on the left, consisting of metallic stent graft rings sutured to a synthetic graft, is placed transluminally along the longitudinal extent of the thoracic aorta, thereby creating a homeostatic seal, excluding the aneurysmal or the disease proportion of the aorta from high pressure blood flow. Now, the use of standard TVAR devices is not suitable for those diseases which encroach onto the aortic, the arch segment, or those of the ascending aorta, simply for the reason that we would end up blocking the supraortic branch vessels, which perfuse the arms and the brain, if using a standard device. So, to treat these patients, surgeons have augmented traditional techniques and have introduced a new procedure known as chimney TVAR, which is illustrated here. Now, in this procedure, devices are deployed two devices rather are deployed in a manner such that they lie parallel to each other or in a so-called nested configuration. The main endograft serves to exclude the disease proportion of the aorta, whereas the secondary or chimney grafts are placed within the supraortic branch vessels to maintain that supraortic branch perfusion. Now, on the bottom right, what we see is this idealized kind of configuration of what this device, what these devices might do in vivo throughout the cross section. What we see is a chimney device, here shown as in a perfectly circular cross section, and a main graft device conforming perfectly to the wall and to that chimney stent. But what we can also see here is the formation of these gaps which form in between the aortic wall, the main graft, and the chimney graft. And these gaps are called gutters. And it's within these gutters that blood can continue to flow and keep that aneurysmal proportion pressurized, which may lead to adverse events later on after treatment. So the questions that we're really interested in here is, how do these devices interact, and do they interact in a way which the illustration I just showed kind of depicts? And if we understand how these devices interact, can we understand or kind of predict if these devices will be used in an effective way? And we can kind of look at things in either a short-term or a long-term perspective. In the short-term, kind of addressing, do those gaps or gutters form? Do the chimney grafts undergo any degree of compression, which we might be worried about for blocking blood flow, for example? And then in the long term, we can look at things like excessive arterial loading and things like device durability and integrity. And the key factor when we're considering chimney TVAR is that these devices are used in an off-label manner. That is, they're being used outside of their instructions for use in a way which they were not intended to be used. Okay, so a point of departure, where do we begin to kind of start studying these things and understanding how they interact? Well, computational frameworks are very good because they allow us to kind of bypass any kind of complicated experimental techniques or using real live devices, which can be difficult to obtain and are very expensive. So numerical techniques offer us kind of at least a place to start, if you will. Okay, now what we've developed here at Oxford is kind of a numerical toolbox for analyzing stent-graft devices. And we've done this in a kind of a bottom-up approach where we start with the material models and then build things up incrementally in an order of increasing complexity. So to begin, we look at the material behavior of each of the individual device components in the stent-graft artery domain. We then incorporate those material models and build up our simulations to study things like stent response to loading and what a device might undergo during packaging into one of those delivery catheters for minimally invasive surgery. And then again, moving kind of towards something which is towards increasing complexity, we can add the graphs to the simulations and begin to understand what does changing the stent design do to graft behavior during bending. And finally, what we can do is perform deployment simulations into arterial segments and begin to understand how different oversizing configurations and different device designs might have on arterial wall stresses. Okay, now what we've done is we've extended this kind of this kind of baseline and simplistic framework into something that's relative to investigating parallel endografting. And what we see here is two configurations of two different main grafts and what the resulting cross-section looks like if placed in parallel with a equivalent chimney graft. And what we can do within this computational framework is quantify things like, chimney graft compression. Now we can see it doesn't have that perfect circular cross-section. We can also identify and quantify gaps which form and begin to assign specific values to those gutters. We can also look at things like graft infolding, which may influence blood flow flowing through the lumen of a device. Finally, given the flexibility of the computational tools, what we can do is we can do comparisons and look at how different devices might behave in different scenarios. And what we're seeing here is exactly that, the cross-section of two different scenarios at different levels of device oversizing. And we can start to see some very compelling differences between different device types. Okay, so how do we know that these observations are correct? Now, computational modeling is playing an increasingly central role across the healthcare sector. Now, before we use these computational models to make any meaningful conclusions, we need to be confident in their correctness, if you will. And so to do that, we're turning towards our experimental techniques. Now, in the first instance, what we need to do is we need to validate our FEA results. And to do this, what we do is take an experimental setup which we can easily and faithfully reproduce in a virtual environment. And those who are familiar with stents and stent grafts will probably know about radial force testing. And that's what we're doing here. So to begin, we take our stent sample. What we do is we take a micro CT scan to obtain its geometry, and then using scan IP, what we're able to do is take that micro CT data and build up a very high resolution, accurate reconstruction of the stent device. Now, based on the high resolution CT reconstructions, we can compute and export the device center lines from scan IP. And that's what we're seeing here on the left-hand side. Now, using that center line data, we can develop and build a very good mesh to do our finite element analysis. And what we're seeing here is the so-called radial crimping experiment in a virtual environment. Now, what does that benchtop testing actually look like? Well, what we have here is our radial force tester inside a temperature-controlled chamber. Now, owing to the availability of the test system, which we used during the aforementioned validation work, we extended the protocol to study parallel endografting of various devices and device configurations, noting here that we also employ a video system to capture mechanical deformations. And just as a quick note on the side, the specific devices that we used in this study, for the chimney grafts, we had both self-expandable and balloon expandable chimney types. And we also had two distinct types of main grafts, one which consists of a series of stents, while the other has a single helical wire along its length. Okay, so what do these experiments look like? Well, on the left, what we see here is the radial loading and the radial deformation of two self-expandable stents placed in parallel. And then the next two videos we see here is the positioning and deployment of a balloon expandable chimney stent in parallel to a main stent graft, and the resulting unloading profile of that configuration. Now, using that video data and some subsequent image processing, we can distinctly identify key regions of interest and quantify things like gutters, chimney graft compression, main graft infolding, and things like that. Given the fact that we performed the testing in a radial force tester, we can also characterize the radial force data associated with different configurations. And what we can see here is some very distinct differences between different device types and configurations, which might be experienced in vivo during parallel endografting. Okay, so the next question now is, can we elaborate from these bench tests into something which is more clinically relevant to the clinical scenario? And what we've done is implemented a simulated use vascular model under physiological conditions. What we have here is a silicon vascular model, including the ascending aorta, the arch, and the descending aortic sections of the thoracic aorta. We've coupled that with a pulse duplicator, which replicates the left heart function. Using that setup, we've performed our simulated use chimney T-bar. And what we've done is we've taken this setup and taken it to the hospital where we can perform equivalent ECG-gated CT scans to really look at specific phases of the cardiac cycle. So here we see the realization of the setup at the John Radcliffe here in the hospital, with the video on the right demonstrating the pulsatile nature of our setup. In these particular experiments, what we were looking at was double chimney endografting, where we place two chimney endografts rather, one into the brachiocephalic trunk, the other into the left common carotid artery, and we place those in parallel to our main thoracic stent graft, which is deployed in the ascending proportion of the aorta and runs down to the descending proportion. Okay, now in this set of experiments, we actually conducted two double chimney T-bar procedures with either balloon expandable or self-expandable chimney stents. On the left, we see a typical CT reconstruction, and for this specific image, we're looking at the balloon expandable configurations. And this is the sort of image which clinicians will encounter on a daily basis during patient follow-up. Now, what we see here on the video on the right illustrates the use of scan IP operating on that same data set. In this instance, we're really interested in pulling out some additional engineering-based data. And specifically, what we can look at is segmenting things like the silicon arterial wall, the main graft, the chimney grafts. And using a workflow developed for this particular study, we can identify and characterize gutter formation within the dynamic model, which is what we're seeing here in green. Now, what does this assessment really look like? Well, on the left, we see that we can identify clearly 4 unique gutters that differ not only in their location, but also in their volume and their shape. On the right, we can see the detailed analysis of a single gutter comparing the end diastolic and peak systolic phases of the cardiac cycle. Now, by overlaying the gutters, we can get an idea of the dynamic nature of the model. By co-registering the gutters, we can obtain a qualitative visualization of how these gutter volumes are changing due to diastolic and systolic pressures. And then by performing a gutter surface deviation analysis, we can actually quantify exactly how these gutters are changing throughout the course of the cardiac cycle. Now, just a couple quick notes on this slide here. All of this work was performed in ScanIP. And what we found particularly of use when working between the engineering departments and the hospitals were the tools that ScanIP kind of offered us, and in particularly the ability to create, for example, 3D PDFs, which we could share very quickly with the clinicians and could give them a really clear idea of exactly these gutter shapes and how these gutters change over the course of that cardiac cycle, as I mentioned. And just another quick note is what we're seeing here is, as I mentioned, a single gutter, but what we're looking at is from a view, or rather as viewed from the anterior surface, posterior surface, and viewed from the inflow of that gutter. Okay, so computational experiments really allow us, or offer us rather, the ability to investigate interactions in detail, which aren't quite possible with conventional experimental techniques. So when we're really kind of asking ourselves, can we characterize physiological phenomena? What we're doing now is moving away from those experimental methods and moving back to some finite element analysis in what we're calling realistic simulations. As part of a larger international collaboration known as the Living Heart Project, which was initiated and is run by Dassault Systems, here at Oxford, we leverage what is referred to as the Living Heart Human Model to study chimney T-bar. Now, on the video on the left-hand side, we see the Living Heart Human Model. Now, this model is a dynamic representation of an adult male human heart whose response is governed by realistic electrical, structural, and fluid flow physics via a sequentially coupled electrical mechanical analysis. Now, using this model, we can perform device deployments. What we can do is, as per the original simulations, we can look at things like strains within the device and how the device interacts with the arterial wall. Crucially, we can extend these parallel endografting cases and investigate how devices interact over the course of cardiac cycle. Now, using the Living Heart Human Model, we can assess the influence of various device combinations, and in this case, some generic devices. Again, we can characterize significantly different post-deployment configurations dependent on device type. Using realistic simulations, we can study how the configurations changes over the course of the cardiac cycle. Moreover, we can compute specific contact force interactions, which we can't do experimentally. And perhaps what is most significant is we have the ability to compute how parallel endografting increases aortic wall stresses. Okay, now, in summary, what we've seen here is that structural interactions during parallel endografting are significantly affected by both device design and configuration. We see things like increased aortic wall loading, likelihood of endoleak, and increased device deformations are dominated by the intrinsic properties of the endografts. Perhaps most interestingly, and perhaps most relevant to the clinical community, is that the phenomena that we're seeing during chimney T-bar are both spatially and temporally dependent. Now, to close, a bit of a general outlook. What we're seeing now is an increased off-label use of standard devices for managing complex aortic disease. And what I had alluded to earlier in the talk is that devices are being used in this in a manner which is outside their instructions for use. And so what we can do is we can use these computational and experimental tools to really identify these optimal device configurations for which no consensus exists. Now, for the device manufacturers perhaps in the audience, what we see here is a real opportunity for next generation TUR devices to better accommodate parallel endografts so that surgeons have more confidence going into these procedures to minimize the risk of adverse events such as gutters and device compression. Okay, so this work really could not have been done without the help of a number of individuals who I want to thank here. And of course, has been made possible by a number of organizations who have either donated devices, access to software models, and experimental resources. So a big thank you to both the people and organizations involved in this work. And with that, I want to say thank you to all of those online who've been watching, and I'm happy to take any questions or comments or discuss any points you have now, or if you're unavailable to get through, feel free to drop me a line via email down here on the right-hand side. So thank you.
Great, thank you, Jake, for that very, very nice talk and nicely presented presentation. Okay, so we will be doing a few questions in a minute. We'll pick a couple of questions from the ones that have been submitted. So do you ever think if you've got a question you'd like to ask us live now, please just type those into the questions toolbox. If we get too many, we will get back to you via email if we're not able to get to your question today. Just while we wait for those questions to come in, just want to say thank you again to Jacob for participating in this presentation and for the time he's invested to help prepare the presentation and provide the talk today. So from here, we'll just do a few questions. If you're interested in trying the software at any point, please visit our website to request a trial. And with that, you get full technical support to help guide you through the introductory process of learning the software and processing your data. Okay, so we've got a couple of questions coming in. So the first one we've got here, Jacob. So what do you see going forward as the next important step in this research?
Yeah, great question. So I think this research, from our end anyways, was quite exploratory. So kind of proof of concept from our side, you know, collaborating between engineering and in the hospital. And what we were really focusing on here were those structural interactions. And although, in the in vitro model, rather, we had things like flow and a pulsatile system there. What this study or in the sense where it's limited is the specific fluid flow aspects associated with gutters and endoleak and things like that. So I think really the next big step for this research is definitely looking at exactly the fluid flow phenomena through those gutters and what constitutes a dangerous gutter versus one which might be considered to be safe or okay in the short term. So I think definitely what we've done here is like the groundwork for some good structural analysis. And I think the next big thing is going to be taking this work and extending it by adding some good work around the fluid flow physics specifically.
Great, thank you for answering that question. We've got a few questions here that have come in on the silicon arterial model. Basically how you made it, how much does it cost, basically a bit of background on that. Do you want to maybe spend some time talking about that?
Yeah, so great question. We were very fortunate to work with Elastrat. And so Elastrat, we worked with them to kind of use one of their off-the-shelf models. And so what I'd highly recommend to do if you're really interested in some really good high-quality models, ones which are readily available, is please get in touch with them. I think they provided us with a really great service to get that. The particular model that we used was an off-the-shelf version. It's based on real anatomy, yeah, nothing too fancy. But if you're interested in custom models, I'm sure that they could help you out with that. The one thing that I will mention regarding the model itself, and what I didn't showcase here is again kind of going back to ScanIP. What we were able to do is kind of quantify very, very well using the wall thickness tools exactly how the wall thickness changed throughout that model, and it really helped us with the analysis to kind of identify, is the model physiological when used in kind of parallel with our pulsatile flow system. And so using ScanIP, doing the wall thickness analysis and various measurements, we were able to characterize it really quite well.
Great, thank you for answering that, and also thank you for referencing how you kind of checked on the thickness as well in the software. So I think we've got through, I think we had quite a lot of similar questions, which I think you've kind of covered with that last question. There was quite a few on the arterial model. So probably best now if we just refer back to your contact details. If someone's got a question for you afterwards, I think it's on the bottom right of the screen, your email address. So we'll probably finish up there. I think that we've got through all the questions. I think there might be one more coming, but I'll just keep talking for now. Just to mention, we have recorded the webinar today. You'll get a link to download this in the next couple of hours or within a day at least, I would have thought, to give you a link to download it and maybe share it with colleagues if needed, or you can find it eventually on the website as well. Okay, I think that's everything. Just double checking the list of questions again. And I think we've got through all of them. Seems everyone was kind of very keen to learn more about the arterial model, it seems. But yeah, thank you, Jacob. Thank you very much for the very nice presentation and slides and talking us through your research. We were very pleased to hear how Scanopy has helped out with that research.
Thank you very much.
So yeah, thank you from myself and Jacob. If you've got any final comments.
No, thank you. If you have any other further questions, please feel free, as Ross said, to get in touch with me directly, and I'll be happy to kind of answer any more questions I can there.
Okay, great. Thank you, Jacob, and thanks for everyone who's listened in today, and thank you for the people who've answered the questions. Have a good rest of the day.